Okay, so we're gonna start the talk of today. We have invited today Jack Bronk, and he's gonna present him. He's a professor in industrial microbiology and the head of the department of biotechnology in the Technic University of Delft. He has been working for a lot of years in the understanding of and the improvement of the microbial performance in industrial processes. And he has used quantitative physiology, genomics, synthetic biology, evolutionary engineering, as his most part of the work. He has been published more than 270 papers, and he is inventor of more than 25 patents. He has been leading the Krieber Center of Genomics for genomics of industrial fermentation in Delft. And he has obtained a lot of awards because he has been publishing key works related to microbial behavior in the last years. And finally, he has been elected as a member of the Royal Netherlands Academy of Science. And from a personal point of view, for me, it's a pleasure to have him here because I was visiting almost 10 years ago, and I could see how the level of high level of and quality of the scientists that is done in the Delft department. So for me, it's a real pleasure that he has some time for us. And today he's gonna talk about identification and elimination of oxygen requirements in yeast. So Jack, thank you.
Thank you, Roberto, for raising the bar to extreme levels. I'll try to live up to that. It's a real pleasure to present something to a group of very high-level scientists. I'm sorry I couldn't come to Valencia due to all the restrictions. Very fond memories of a previous visit. And as I say, it's fun to share some very recent results of our research on yeasts and oxygen. And what I'll do is to give a rundown of some results from a wonderful chance I had to dig into this subject, which is very close to my heart, with a 5-year ERC grant. This weekend, by a coincidence, I have to write the final report. So a lot of it is fresh from the needle. So this project set out to identify and then to eliminate oxygen requirements in yeasts in general. And I'll explain that in a bit more detail. But the focus today will be, for a large extent, on sterol synthesis. And I'll explain a little bit later why that became a very intriguing theme. But first, when we talk about oxygen requirements, intuitively, we think about respiration. And that's definitely also relevant for yeast. The large majority of yeasts are able to respire sugars, and sometimes other substrates, to water and CO2, and in doing so conserve free energy in the form of ATP. But also a very, very large majority of yeasts is simultaneously able to ferment sugars to ethanol and carbon dioxide. I'm not telling you any secrets so far. And the yeasts that can ferment invariably do so under oxygen-limited conditions. Now, if you look at the alcoholic fermentation pathway, this is very classic biochemistry. It's first redox balanced, and secondly, it results in the net generation of ATP. So the ability to ferment sugars to ethanol implies that facultatively fermentative yeasts should be able to generate ATP in the absence of oxygen, in the absence of respiration. And from that, we might easily conclude that then probably this whole large group of over 1,000 yeast genera that's able to ferment will also be able to grow in the complete absence of oxygen. And my ERC project started out with the observation that this is absolutely not the case. In fact, only very few yeast species can grow in the complete absence of oxygen. This inability of the large majority of yeasts to grow anaerobically has been attributed to mostly unidentified requirements for molecular oxygen in biosynthetic processes. And there's a quite impressive list of biochemical processes and reactions that have been implicated in this context. Redox cofactor balancing, pyrimidine biosynthesis, synthesis of cofactors, of enzymes, heme biosynthesis, ribonucleotide reductases. And today I'll start with the synthesis of lipids, and in particular of unsaturated fatty acids and ergosterol, the main sterol in yeasts. And there are also some aspects of this that we studied intensively in the project that I will not go into. For example, we engineered a pathway that enabled anaerobic biotin biosynthesis into Saccharomyces cerevisiae and thereby made anaerobic cultures oxygen independent. But let's start with fatty acids and sterols. There are two very famous and often cited papers from the early 1950s that have resulted in something we might call a dogma in yeast physiology. And I don't think, looking back, this was necessarily the intention of the authors. And I'll try to come back to that. But anyhow, the dogma was that any yeast that is able to grow in the absence of oxygen requires supplementation of growth media with at least one unsaturated fatty acid, and a source of sterols. And it's also known that these factors are relevant in industry, and then it concerns Saccharomyces yeasts, either Saccharomyces cerevisiae or hybrids used in brewing, wine fermentation. In these anaerobic processes, depletion of sterols as they become deeply anaerobic due to CO2 production or mixing in these systems causes so-called stuck fermentations. At the outset of the project, the yeast contain some sterols and unsaturated fatty acids, but as the process becomes strictly anaerobic, these run out and the fermentations can just stop. And this is often countered by a very intensive short aeration phase prior to the anaerobic fermentation to allow the yeasts to build intracellular stores to compensate for an inability to make these two essential compounds. The oxygen requirement for synthesis of unsaturated fatty acids in yeast, in Saccharomyces cerevisiae in particular, is very well understood. In contrast to many prokaryotes in which double bonds are introduced during chain elongation, yeasts first make a saturated acyl-CoA and then introduce the double bond, desaturate the chain in an oxygen-dependent reaction catalyzed by the desaturase Oli-1. And so we have a very clear link between biochemistry and a biosynthetic oxygen requirement. So when we started setting up systems for checking and ultimately eliminating these oxygen requirements, we took this as a control experiment. And I'll try to explain step by step how we did that. First, in order to say something about requirements, biosynthetic requirements for oxygen, it's very important to deplete any intracellular stores. So in this control experiment, we looked into requirements for unsaturated fatty acids, which we had in the form of tween 80, an ester of oleic acid, and ergosterol, the main form of sterols in yeast. And the first thing we do then is to have an anaerobic growth cycle in an anaerobic chamber in the absence of both requirements to allow the culture to deplete intracellular reserves, and growth stops here quite early. And then we confirm that when we add in one culture both of these anaerobic growth factors, we get fast growth. And we did, so that was promising. And when we leave out both, we see virtually no growth. If we only take out the ergosterol, we see a little bit more growth. There's still oleic acid present here. And this we attribute to a little bit of oxygen leakage into the system. And indeed, when we do analysis of the lipid fraction here, we see tiny amounts of sterols. Yeast has an enormous affinity for sterols, Saccharomyces cerevisiae, for oxygen in sterol synthesis. Saccharomyces cerevisiae does at least. And it's difficult to eliminate the last traces of oxygen. But what really surprised us, and which appeared to contradict this old dogma in the literature, was that we got quite substantial and fast growth in the absence of a source of unsaturated fatty acids, in the presence of sterols. And here we got very, very suspicious, because reading the literature on these oxygen requirements is difficult, because there's always the uncertainty whether there's not some oxygen contamination in the system. But this was a completely unexpected observation. Fast growth in the absence of unsaturated fatty acids. So what's going on? Just to illustrate the technical challenge here, the oxygen requirement for making sterols and unsaturated fatty acids in Saccharomyces cerevisiae is about 100 micromoles per gram biomass. And we know that Saccharomyces cerevisiae can still synthesize ergosterol at nanomolar concentrations of oxygen in the liquid phase. So keeping out oxygen in anaerobic growth chambers, which we use quite intensively, and especially in bioreactors, requires a lot of measures. And I won't go into this in depth. We summarized our findings in a recent protocols paper, if you're interested in the subject. And as I already indicated, it's also important to eliminate intracellular reserves by, let's say, a carryover culture in which we allow the cultures to deplete any such reserves. But even after taking these precautions, we felt very uncomfortable about this unexpected growth in the absence of unsaturated fatty acids. So what we did was to eliminate the gene encoding the desaturase to be absolutely sure there could be no de novo synthesis. And then we repeated the experiment a number of times with subsequent transfers to fresh medium, and the cultures kept growing. There was sustained growth in the absence of unsaturated fatty acid supplementation. And we saw that in the absence of supplementation of oleic acid in this case, there was a change in the average chain length of the saturated fatty acids, which makes sense because shorter saturated fatty acids, in the same way that unsaturated fatty acids do that, make membranes more fluid. So this is a sort of logical compensation process. Now, some of you may wonder, hey, we see a declining growth rate here. We have confirmed that this is due to the moment at which we transfer the cells. So these cells are not very happy, they're less robust. And if we leave them in tween 80 supplemented cultures, and if we leave them too long in stationary phase, growth in the next cycle takes more time to kick off, but the cells do keep growing. So that was, we sometimes jokingly say, killing a dogma, but if you go back to the old literature of Andreasen and Stier in the 1950s, there are already indications for some growth in the absence of unsaturated fatty acids, but the field, including ourselves for many decades, has kept referring to these studies as evidence for an absolute requirement for oleic acid. So it's important to go back to the old literature now and then. And I'll have an even more spectacular case of that later on in the presentation. Well, the other dogma is that sterol synthesis is needed for anaerobic growth of yeast. And looking at the pathway for ergosterol synthesis, this makes perfect sense, because making 1 mole of ergosterol via a very strongly conserved pathway requires no fewer than 12 moles of oxygen. And there's no evidence in living organisms or in the archaeological record for anaerobic synthesis, oxygen-independent synthesis of sterols. So that seems to be a fairly solid dogma. And this does not mean that all eukaryotes require sterols for anaerobic growth. And the reason for this is that some fungi have found a way out. This is interesting. The sterols are essential in many eukaryotes for membrane structure and function, also in industrial contexts. But sterol synthesis is also an important target for many antifungals. So understanding here what's going on is fundamentally interesting, but it also has some applied aspects. So I already said, well, there are some fungi that do not need sterols. In the same way that in the Asterix stories, there's this tiny village that behaves a little bit peculiar. There's a group of fungi called the Neocalamesticomycetes. Nice name to practice. And these deep branching anaerobic fungi have found another solution to anaerobic growth in the absence of sterols. And what they do is to not make ergosterol the main sterol in fungi, but they make a sterol surrogate, a pentacyclic triterpenoid that in terms of structure sort of vaguely resembles, well, a bit more than that, ergosterol. We isolated a strain of these Neocalamesticomycetes from cow dung. They're very difficult to keep alive in liquid nitrogen or in culture collections. So postdoc took on the brave task of going to a farm in the neighborhood, doing some unspeakable things with cow dung, and came up with a pure culture. This is the pathway for sterol synthesis again, and you see the typical flat planar structure of ergosterol. And what these fungi do instead is take the precursor of sterols, squalene, and do one cyclization reaction catalyzed by a squalene tetrahymenols cyclase, and they make this compound here, tetrahymenols, which is this pentacyclic structure. And you can roughly see the similarity of sterol and tetrahymenols based on these 3D representations. So in these anaerobic fungi, in these Neocalamesticomycetes, based on sequence, it's clear that this has been obtained, the gene encoding this tetrahymenols cyclase has been obtained by horizontal gene transfer. But from that, just from observing that the gene resembles genes in other classes of organisms, we cannot conclude that just the acquisition of such a gene is sufficient to confer the inability to grow in the absence of sterol supplementation. It might require a lot of additional modifications, and these anaerobic fungi are not genetically tractable in the sense that we cannot go in there with CRISPR or other molecular genetic techniques yet. So we decided to use Saccharomyces cerevisiae as a test bed here. And the question was, is the mere expression of such a tetrahymenols cyclase, squalene tetrahymenols cyclase, sufficient to enable Saccharomyces cerevisiae to grow anaerobically in the absence of sterols? So the question was really very simple. If we stick in squalene tetrahymenols cyclase gene from Tetrahymena, in this case, the organism in which it was first discovered, do we first get synthesis of this sterol surrogate? And secondly, does it enable anaerobic growth in the absence of sterols? And again, to be sure that any observations would not be due to oxygen leakage into our cultivation systems, we tested this in a strain in which we eliminated sterol synthesis by knocking out the gene encoding the first enzyme in the pathway, an ERK1 mutant. The results are summarized here. It's quite a lot of work in one slide, but indeed, we were able to demonstrate that a strain incapable of synthesizing ergosterol, or any sterol for that matter, but expressing this tetrahymenols cyclase, was able to grow anaerobically in the complete absence of oxygen and also exhibited sustained growth. And this we took to extremes because there's some literature on sparking effects of very, very small amounts of ergosterol. And we did serial transfer experiments in bioreactors with this strain until we could, with a high degree of confidence, calculate that there was not a molecule of sterol left in any of the cells in the culture. And so a large number of serial transfers. And when we looked at the lipid composition, we could indeed see that this strain, grown under anaerobic conditions, did not contain any sterols, but did now contain this sterol surrogate tetrahymenol. So by doing this, we in a way simulated the horizontal gene transfer event that's being proposed to have occurred in these anaerobic fungi and showed that it immediately confers a selective advantage in sterol-limited anoxic environments. We also saw, and I've not gone into any detail here, that these yeast cells were not exactly happy in this situation, in the sense that their specific growth rate and also their ethanol tolerance, for example, appear to be lower than that of wild type strains. For the specific growth rate, we saw that. For the ethanol tolerance, we inferred it. And we use this a little bit further in follow-up research to look at other adaptations of these deeply anaerobic fungi, of these neocalamesticomycetes, to anaerobic growth. And we were able to show by functional complementation of Saccharomyces cerevisiae mutants that during their evolution for growth in anaerobic environments, these fungi also acquired functional pathways involved in the synthesis of NAD and NADP, so redox cofactors, and for oxygen-independent synthesis of pentophenate and from there coenzyme A. So this approach of using cerevisiae as a test bed proved to be a very productive and nice way to study these difficult to handle fungi. I already hinted a little bit at industrial relevance. Working in a biotechnology department, in industrial microbiology group. And from the outset in this project, we had one specific target. For large-scale bioethanol production, and currently not we, but industry makes about 100 million tons of ethanol by yeast-based fermentation, all with Saccharomyces cerevisiae at the moment. For these processes, there are very important economic incentives to do it with more thermotolerant yeasts. And this is because of better heat economy, less cooling costs, etc., but mainly also to reduce enzyme dosages, polysaccharide hydrolyzing enzyme dosages in these anaerobic processes. And the latter holds especially when industry wants to deal with with more complicated lignocellulosic substrates that require a lot of cellulases, hemicellulases to chop up the sugar polymers. One yeast we had our sights on is Saccharomyces marxianus, which has been dubbed the fastest growing eukaryote on Earth. Still, I don't know if it still holds that record. It can grow very fast, it can ferment very fast, but it cannot grow in the complete absence of oxygen. And Wijp Dekker, one of the PhD students on the project, took on this challenge and first performed an intensive physiological study to see if we could get indications where this oxygen requirement might be. If we grow Saccharomyces cerevisiae at very, very low oxygen supplies in chemostat cultures, it's difficult to get the system really oxygen limited. And the oxygen contamination in these reactors is so easy that even under stringent conditions and very anaerobic, and just adding tween 80 here, so leaving out the sterols, the yeast still manages to eat up most of the glucose. This shows the glucose that is not used in the cultures. But if we do the same with Saccharomyces marxianus, it leaves an enormous amount of sugar. And one thing we observed here was that if we edit, so if we leave out both tween 80 and ergosterol, a lot of the sugar is used. And we expected that it's, sorry, I'm telling you the wrong way around, so I'll keep it short. From comparing cultures with different supplementations, we saw that adding sterols or not, did not really affect the amount of biomass that the yeast could make or the amount of sugar it could consume in these cultures. In other words, it seemed that Saccharomyces marxianus was unable to use sterols we supplemented, because if that would have helped, we would have expected a difference here in biomass and sugar concentrations. And then when we looked deeper into its genome and into gene expression, we also found there are no clear orthologs of the two genes in Saccharomyces cerevisiae that encode sterol importers, OS1 and PDR11. We just cannot find anything that clearly resembles these transporters in Saccharomyces marxianus. We followed up, Wijp, the PhD student, followed up by uptake experiments with a fluorescently labeled sterol analog, MBD cholesterol. If we add this compound to Saccharomyces cerevisiae, we see a clear increase in fluorescence of the cells. So in the flow cytometer, the peak here shifts to the right, indicating more indicating uptake. Interestingly, in a mutant lacking the transporter, we also see a more moderate shift to the right. And this is caused by the fact that sterol uptake in Saccharomyces cerevisiae does not just require a membrane transporter, but also cell wall proteins that bind the sterol. And what we see here is fluorescence in the cell walls of these proteins that may be involved in transfer of the sterols across the fairly hydrophilic cell wall to the membrane transporters. But in Saccharomyces marxianus, nothing happened. So there was no shift of the peak. And this indicated that Saccharomyces marxianus really lacks a sterol transport system, a functional one. Now, due to the involvement of cell wall proteins in sterol import by Saccharomyces cerevisiae, this is not so easy to reconstruct in K. marxianus. So we chose for a different approach. We just discussed this sterol surrogate, tetrahymenol, and to test the hypothesis that sterol uptake is really the key limiting step that prevents Claphomyces marxianus from growing anaerobically, we expressed the tetrahymenol cyclase, now not in Saccharomyces cerevisiae, but in Claphomyces marxianus. And we got lucky here. The hypothesis turned out to be correct. So expression of this tetrahymenol cyclase in Claphomyces marxianus resulted in strains that were able to show sustained growth upon repeated transfer in anaerobic cultures under anaerobic conditions. And in the absence of ergosterol, and also in its presence, because it doesn't matter if you add it to Claphomyces marxianus. And we were able to show that the strains indeed also produced tetrahymenols. So that seemed almost mission accomplished. It was a very cool result, but in more ways than one. This was all done at 30 degrees, and our whole motivation to look into Claphomyces marxianus was its high temperature maximum. Strains didn't immediately grow very fast at high temperatures, and so at the high end of its temperature range, but by serial transfer in bioreactors, fill and draw system, where we kept them growing for a large number of generations, we were able to obtain strains that grew anaerobically without sterol in the biomass at 45 degrees centigrade. And again, these are not yet strains that could be applied in industry, not extremely robust yet, but we have a starting point here to work towards, for the first time, really anaerobically performing yeasts for bioethanol production. So we were very happy with this result. So conclusion, absence of a functional system for sterol uptake is decisive for the inability of K. marxianus to grow anaerobically. But to get to industrial strains, there's still a lot of work ahead to make strains more robust. And you might conclude from this, from the experiments on Saccharomyces cerevisiae and Claphomyces marxianus, that these surrogates are really poor substitutes for sterols. Cells grow a bit slow, they're not so robust. So is this a dead end? And I must admit, we sometimes wondered ourselves, although I was optimistic, I still am. And then something really nice happened in our research. Based on some observations from our own research, we went back to the old literature and came across this 50-year-old publication by Bulder, famous yeast researcher from our country, from the Netherlands, who already in 1971 published that there's something very peculiar about the fission yeast Saccharomyces japonicus. And what he reported was that whereas Saccharomyces cerevisiae, when grown in chemostats under anaerobic conditions, requires sterol supplementation, Saccharomyces japonicus does not. And for someone who's been working on yeast metabolism and oxygen requirements for quite some time, it was quite shocking to me that I'd never really studied this paper. I had not read it, to be quite honest. But also that it's only very rarely cited in the field. It seems that this has been overlooked. Now, we were skeptical about our own experiments, very almost paranoid about oxygen leakage into our cultivation system. So the first thing we did was to check whether Bulder got it right and whether this was not an artifact of oxygen influx into the culture. Working with this yeast required some very special measures because it exhibits light-induced sporulation. And this messes up any quantitative growth experiments. So Jonna Bouwknecht and Sanne Wiersma, the PhD students working on the project, nicely built a dark room around our anaerobic chamber setup which was very spooky during COVID times because the labs were already empty, and then all of a sudden you'd hear voices coming out from underneath this shroud. But it worked, and they did some amazing experiments that completely confirmed what Builder had reported. And here it's sufficient, I think, just to look at the open squares, which indicate cultures that contain neither tween 80 nor ergosterol. And of course, there's the combinations. But Schizosaccharomyces japonicus really does not care. It grows just as fast without tween 80 and ergosterol as it does with these two compounds. And this is, to our knowledge, the only yeast that exhibits this behavior. And we were that curious how this could be. For the UFAs, perhaps the explanation was not so tremendously shocking based on what we saw in Saccharomyces cerevisiae. We see an adaptation that is very similar to what we saw in Saccharomyces cerevisiae. The only one mutant grown anaerobically. Now in Schizosaccharomyces, we also see that the fraction of short-chain saturated fatty acids increases as we leave out oleic acid from the medium. We also see that under aerobic conditions, this yeast is able to synthesize unsaturated fatty acids itself. So we have a very peculiar adaptation here to grow anaerobic growth without UFAs. And in contrast to Saccharomyces cerevisiae, which is struggling a bit when we get it to grow under these conditions, Schizosaccharomyces japonicus appears to be very happy. It grows very fast and to high biomass densities anaerobic. But what's going on with the sterols? Because it seemed extremely unlikely that this yeast would have evolved a completely oxygen-independent sterol synthesis pathway. So we looked at the triterpenoid fraction of the terpenoid fraction of the lipids. And in Saccharomyces cerevisiae, we see ergosterol, which is added to the medium of anaerobic cultures. And we see a bit of lanosterol, one of the first intermediates in the pathway. And we don't see anything of these components in Schizosaccharomyces japonicus. But we do see peaks that do not occur in Saccharomyces cerevisiae, and when we did mass spectrometry on these peaks, they turned out to be hopanoids. So not tetrahymenol, which we might have guessed, and which we thought for a while, perhaps this yeast is able to make tetrahymenol. It's not, but instead it makes a different class of pentacyclic triterpenoids. So hopanoids like tetrahymenol are pentacyclic triterpenoids. And they occur a lot, especially in prokaryotes, where they have a role that's very similar to that of sterols. Control membrane rigidity, influence membrane permeability, positive way. And these compounds can be formed like tetrahymenol in a single cyclization step, starting out from squalene. And here we have these compounds, and we were able to identify a number of them, including diplopterol. In the meantime, of course, we were already looking into the genome sequence of Schizosaccharomyces japonicus. And there we found a very, very strong homologue of genes from acetic acid bacteria in particular that encode the squalene hopanoid cyclase. And it's known for these enzymes that from one substrate, squalene, they're able to generate a number of different products, different hopanoid products. And that was also in line with our mass spec analyses. So this indicated a very clear, this provided a very clear indication that Schizosaccharomyces japonicus acquired a bacterial gene encoding such a hopine cyclase from acetic acid bacteria. And for me, as a long-standing Beatles fan, it was very nice to see that both these organisms have been isolated from strawberry fields. So again, the question was, is this enough? Is it just a correlation, or are more changes required in the genome to make this system work? And to answer that question again, we resorted to expressing the gene in Saccharomyces Cerevisiae and found that indeed, in the absence of sterols, expression of this squalene hopanoid cyclase gene from Schizosaccharomyces japonicus in Saccharomyces Cerevisiae, we enabled S. cerevisiae to grow anaerobically. But I must say that the growth rates were less impressive than they were with tetrahymenol. So there was clearly a difference with the reference situation, reference strain, where the growth rate was very slow. But a clear indication that, like we saw for tetrahymenol, the acquisition of this gene does immediately confer an advantage under anaerobic conditions in presence of very low sterol concentrations or complete absence of sterols. So this turned out to be a new, previously unknown adaptation of eukaryotes to anoxic sterol limited environments. Very probably a case of horizontal gene transfer. And as I'll show you in a minute, if we still have a little bit of time, this is not the only adaptation of Schizosaccharomyces japonicus. It's like these Neocalamasticum mycota, a yeast in this case, that has done some very interesting things during evolution to adapt to anaerobic growth. And Schizosaccharomyces japonicus grows as fast as Cerevisiae in sterol supplemented cultures when japonicus is grown without these compounds. So it appears to be really robust, and it will be very interesting to explore what other aspects, what other characteristics of the membrane are responsible for this fast growth and robustness, because we've not yet been able to achieve that in Saccharomyces cerevisiae. There may also be a medical angle to this, because as I mentioned earlier in the presentation, sterol synthesis is a very important target for antimycotic drugs and to combat fungal infections. And the acquisition of such a cyclase might be an interesting escape mechanism, an interesting resistance mechanism. We haven't seen it so far. An interesting resistance mechanism to escape from these drugs and become resistant. Finally, this is something we published a couple of weeks ago, and I just couldn't resist the temptation to show you that there's more than sterols and unsaturated fatty acids when it comes to anaerobic growth. And I'd just like to show a few slides on something else that I think is really cool, and that relates to pyrimidine biosynthesis. Pyrimidine biosynthesis, irrespective of the organism we look at, this is a key reaction, the oxidation of dihydroaurotate to orotate by a dihydroaurotate dehydrogenase. There are two main classes of dihydroaurotate dehydrogenases, which can be distinguished based on their electron acceptors and structures, but let's focus on the electron acceptors. Now, in the very large majority of eukaryotes, a so-called class 2 DHOD, which uses quinones as electron acceptors, is responsible for pyrimidine biosynthesis. And this quinone-dependent enzyme is linked to mitochondrial respiration, which in yeasts and fungi, almost by definition, makes it oxygen requiring. And indeed, this is one of the processes that's implied in the inability of a large number of yeasts to grow anaerobically. Saccharomyces cerevisiae, which can grow without uracil or pyrimidine supplementation in anaerobic cultures, as belongs to a small group of yeast that have acquired a so-called class 1 DHOD that instead uses fumarate as electron acceptor, and that enables it to make sterols in the absence of oxygen. And for a while, this looked very clear-cut. To be able to make pyrimidines under anaerobic conditions, you need a horizontal gene transfer, if you're a yeast, to acquire such a class 1 DHOD. There was already one puzzling observation from the group of Jure Piskur, who sadly died maybe 7 years ago. Jure was also looking intensively into this metabolic challenge related to anaerobic growth. And he remarked that the yeast Dechra bruxellensis, which is capable of slow anaerobic growth in the absence of uracil or the pyrimidine supplementation, only appears to harbor such a class 2 respiration-linked dihydroaurotate dehydrogenase in its genome. And that messed up an otherwise very clear picture. And the question how this worked was never resolved. And it was also unclear whether this was just a peculiarity of Dechra or whether it might occur elsewhere. So we looked into this with two of the organisms you've already seen, these Neocalamesticomycetes and also Saccharomyces japonicus. And we found that, like Dechra, they also appear to only contain such a class 2 DHOD. And instead of showing you growth curves, etc., I'll just share the main conclusions. When we took the structural genes for these class 2 DHODs from Neos and from Saccharomyces japonicus, we found that they complemented the phenotype of a URA1 mutant of Saccharomyces Cerevisiae. In other words, these class 2 DHODs functioned anaerobically. They shared mutation in the active site, where there's a very strongly conserved serine in this class of enzymes. And these enzymes contained a cysteine there, which in one of the three cases we could correlate directly with its ability to grow anaerobically, but not for the other two. And for the Neocalamesticomycete DHOD, also such a class 2 enzyme, we were able to show that instead of quinones, it uses free flavins, either FMN or FAD as the electron acceptor, which nicely explains why it can function anaerobically both in its natural host and in Saccharomyces cerevisiae. So very interestingly, I thought this is clearly a case of convergent evolution. Three taxonomically phylogenetically very diverse organisms have come up with the same or very similar solutions to changing their dihydroaurotate dehydrogenase and thereby their pyrimidine biosynthesis pathway in order to enable it to grow, to function anaerobically. I'm not going to repeat a lot of conclusions about yeast and oxygen. Perhaps I've done that too much already. But I would like to share, especially here, Granddad talking, especially perhaps with younger researchers, some things I picked up along the way. First, for quite many years, I was quite shy about going for a prestigious personal grant. Basically, the idea was that this is something for others. The terms imposter syndrome may not be entirely alien to me. And what I found is that, and my advice would be, let's phrase it this way, go for it. Don't hesitate. And an important driver for that is also that I found out myself and also saw that with colleagues that there's really intrinsic value in sitting down, reserving some quality time to think up what we would most like to do if we would get the opportunity to work for 5 years on an exciting project with a quite lavish budget to do so. So it's a unique chance if you get it, but also the way there to write the proposal, to discuss it with colleagues, has value in itself, in my opinion. For me, after working for many years predominantly with Saccharomyces cerevisiae and some related yeasts, it was extremely inspiring to bring on these weird anaerobic fungi and work with those in the lab with their genome sequences. A reminder that old literature can be extremely relevant. And finally, the ERC project also enabled me to bring for a number of years a specialized bioinformatics postdoc on board, Raoul Ortiz, now working in Louvain in Belgium. And this added multidisciplinarity in the research group was really an asset. And I've learned from that and will take that forward, hopefully, in my career. Thank you very much for your patience and attention, for your interest in this presentation. Thanks everyone who contributed, especially also the funders, ERC, and some other generous parties. And with that, I conclude this presentation and hope there are some questions.
Thank you, Jack. That was very interesting. Thanks for all the comments on the work that you have done in the last years. That really interesting. And yeah, for the participants, if you have any question, you can write on the chat or just raise your hand and I turn the camera on and you can interact directly. And in the meantime, they ask right questions. I just want to ask 2 details about the work you've done with Japonicus. So they have this analog steroid in the membrane, and you look at the resistance because they grow well, as you said. They are resistant to ethanol, oxidative stress, and at the same level as Cerevisiae does?
Oh, I would not go so far as to say they are as tolerant as Saccharomyces cerevisiae, basically because we did not have the ability to test that. And I was, as I expressed, very happy with this ERC project in terms of timing. It was not a very lucky coincidence to have the last 2 years coincide with a pandemic, but I guess more people have problems with that. So we were limited in our ability so far to follow up. This work has been published very recently also because we had to conclude the PhD projects, but we will be looking into that in the future. But where we see that just the expression of these cyclases in Cerevisiae results in very low biomass yields, and in the Hopin cyclase even in a premature cessation of growth, as in cessation of growth before all the glucose is depleted. And we have good indications that it's ethanol that gets in the way there. Saccharomyces japonicus is able to fully deplete 20 grams per liter of glucose in an anaerobic culture. So it's much more tolerant than a Saccharomyces cerevisiae strain expressing the cyclase. And we would very much like to know why. What is there in the membranes? I guess it must be in the membranes or elsewhere in the cells that enables this yeast to perform so well in the absence of sterols and unsaturated fatty acids.
Right, it must be something. It's not related with the protein in the mitochondria.
Yeah, we don't know, but proteins could be a very important part of the answer here. And okay, mitochondria, but I'll resist the temptation to jump on that straight away.
So and I had one because I have I don't know if I understood that, because in Cerevisiae, when you don't add the low chain fatty acids, and you add sterol, so they can grow anaerobically just fine. But
fine, but they do grow. Yes.
yeah. So your hypothesis is that there are oxygen traces that allows them to make the double bond in the.
No, in the case of Saccharomyces cerevisiae, when we just provide sterols, and we even knock out the desaturase, so the yeast is really unable to make unsaturated fatty acids. We cannot find any trace of them in the biomass. The yeast is still able to grow, and that was very surprising because the field had been almost unanimously accepting that it's impossible for Saccharomyces cerevisiae to grow without unsaturated fatty acids in its membranes anaerobically.
Yeah.
And this shows that that assumption is simply not correct, and at least not in all strains and under all conditions. Now, perhaps the interesting thing is that whereas we see anaerobic growth of these cultures, we have so far not been able to see sustained aerobic growth in the absence of unsaturated fatty acids in the membranes. And that might very well be related also to mitochondrial issues, but we don't know. That's just speculation.
So you're working on this to try to solve the puzzle.
Yeah, right. There's still a postdoc working on some loose ends, or we'll see whether they're loose ends or very nice points for new research.
Yeah, it's a nice question to look into. So there are a few questions. So Sergi asked that the sterols and saturated fatty acid synthesis require iron as a cofactor in oxygen dependent steps. So he's wondering if these novel oxygen independent pathways, these yeasts, require iron or not.
No, the squalene cyclases, to the best of my knowledge, do not have metal cofactors. And I say to the best of my knowledge to leave a little bit of room for doubts, but I'm fairly confident they don't.
Right.
So divalent iron will be available in these anaerobic cultures. It becomes very difficult when trivalent ferric iron is needed for reactions, because that's a lot harder to get by under anaerobic conditions. So, and that is an issue that pops up elsewhere in metabolism with thiamine biosynthesis, for example, iron status. And we didn't really manage to get our head around that yet.
Okay. And we still have one more question about, it's from Amparo that appreciates your interesting talk. And he says that I suppose that the interest in Marcianus is for bioethanol. And the industrial level, they are using anaerobic conditions for production of bioethanol.
The pre-cultivation step, so to generate the seed culture for these processes is not strictly anaerobic. So from the outset of the process, there will be sterols in the biomass. There may also be some plant sterols present in the mesh, in the raw material, but that's very much dependent on the location, the process design, the biomass, the biomass, the sugar hydrolysate that's used. But there are well-documented cases where fermentations get stuck because of a loss of sterols. And one typical thing, it depends a little bit where you look. So in Brazil, it's different from the United States, for example. In the United States, most cultures have to be started with fresh inoculum every time. If we would want to make the process more sustainable by continuous bioprocessing or reutilization, biomass recycling, then this will become a very important issue, because once fermentation kicks in, the CO2 production by the reactors is so intensive, and the area to volume ratio in these huge tanks so small that the system is completely anaerobic. I mean, anyone who's been in a beer brewery and stuck his head in a fermentation vessel will know that it's pure CO2. It just hits you in the face.
So the optimal condition is aerobic, but the volumes make the conditions to be partially anaerobic.
No, the optimal condition would not be aerobic, because that would go at the expense of product yield, because you'd get respiration and
right.
Oxygen requiring reactions running, the risk of contamination would become a lot higher. So stoichiometrically, for some of these yeasts, the ideal situation would be one in which you accurately dose very tiny amounts of oxygen, distribute them over a 1500 cubic meter tank throughout the process, but that is very complicated and expensive.
Right. Sergi, comment again. I read that many industrial applications, they use yeast strain lacking the ergosterol biosynthesis pathway. What can you tell us about that?
I'm completely unaware of it. And given the... personally, I don't think... Well, I'm not saying it's not the case. I'm not aware of it. But what we find is that yeast strains that lack ergosterol have very serious problems with respiratory capacity, so mitochondrial function. And although in the final fermentation process, the anaerobic phase, the fermentation phase, this may not be crucial in all respects, it's absolutely essential for economically viable processes to generate the inoculum for these processes. So the aerobic pre-cultivation steps, and also for strain maintenance. So it may be that there are mutations in the sterol pathway that I can very well imagine, but not that they would be completely sterol negative. And what has been documented by the group of Jens Nielsen, and that's very interesting, it's been known for a long time already that Saccharomyces Cerevisiae is quite promiscuous where it comes to the source of sterols. So Cerevisiae can be grown anaerobically with ergosterol supplementation, but also with cholesterol and other sterols. What Jens and colleagues found in laboratory evolution and reverse engineering sequencing studies, they found that if the sterol pathway, the pathway towards ergosterol, is terminated at the level of fecosterol, the strains become a lot more well, a lot more, 1 or 2 degrees more thermotolerant, which is significant. So that sort of mutations could be interested in industry, but there's always trade-offs, like there will be with these sterol surrogates, that better performance under one condition will lead to some sacrifices elsewhere.
Right. Amparo is commenting also on the discussion that, yeah, in wine, the situation is almost similar because there is aeration, a little bit of aeration at the start of the fermentation. With the ergosterol of the grapes can be enough. So maybe she can talk to you. Wait a minute.
That would be nice. Hi, Amparo. Are you there? Yeah, good to see you. Hi. I cannot hear you yet. You're still muted.
Amparo, activa el micro.
You see, live communication always beats online connections. But we still cannot hear you, Amparo. Sorry.
No, wait.
Yeah. Still indicates you're muted.
And now you can
Now.
listen. Oh, yes. Okay. Only I want to say you hello and thank you very much for this nice talk. And I hope to see you in 1 year we have a positive resolution with the Marie Curie, and we can collaborate. Only.
Super.
Hi.
Great to see you, and thanks for having me.
Okay, bye bye, Jack.
Yes, bye.
And yeah, one more thing. The comment in our Sergi book says that in their hands, only one is an essential gene. I wonder if you have used a yeast genetic background in which this gene is not essential in the absences of unsaturated fatty acids.
What we find is that it's all our indications are that it's essential under aerobic conditions, but not under anaerobic conditions. And this is also why we think the field may have overlooked that under anaerobic conditions, it's not completely indispensable, but that sustained growth is possible.
Mm-hmm.
But to test that, very often people would start by making an aerobic pre-culture. And if that then doesn't grow, it's perhaps not so logical to follow up with anaerobic culture. So that's what we find in the background we use, which is called SENPK. It's used quite a lot for quantitative physiology studies. We haven't followed up in industrial strains or whatever, because we still think that for industrial purposes, something does need to be done to enable anaerobic synthesis of unsaturated fatty acids, and we're working very hard on that still. That's one of the actions of that the postdoc has taken up.
Right. So I think that was all the questions. If there's no any more question, just want to say goodbye to Jack. So yeah, it was really a pleasure to have this excellent talk. Wait, Amparo is want to comment again.
I think it's the applause sign, but or not.
Wait a little bit. No, she's just making an applause. It's not commenting anything. Yeah, because other people, Agustin and Seri also are commenting on that they are thanking you for this excellent talk that you have shared with us. And yeah, I really have to say thanks to save some time for us, because we have enjoyed a lot your excellent science. And yeah, I hope we can see each other soon in your science again.
Yeah, thank you very much. The pleasure and the privilege was entirely on my side. So thanks for freeing up time to listen to some things that are close to my heart. And I hope to see many of you live in the coming time. Stay healthy.
Yeah.
Stay safe.
Same. Thanks. Thank you very much.
Bye-bye.
Bye.